All right, well, hello everyone. My name is Sebastian.
I'm Javion.
In this summer, we worked with Dr. Butcher in the ICR facility.
During our research project, we analyzed protein content from E. coli for further advanced applications.
So what is the significance of proteins? Well, proteins actually carry out most of the functions in all living organisms. Proteins are composed of 20 unique building blocks called amino acids. These amino acids are used by our body to create new cells or repair broken ones.
The ultimate goal of our research project is to isolate these amino acids from E. coli bacteria using the methods we developed. To isolate and identify these amino acids, we need to break down proteins into their individual components using proteases, or proteins designed to break down other proteins.
So how do we actually extract these proteins?
First, we grow our E. coli cells on a petri dish until we see visible colonies. Then we isolate one colony and place it in a special broth that will give them nutrients to grow. Once this broth is saturated with bacteria, we will subject it to high-frequency sound and a lysis buffer, or a special soap, that will break down E. coli cell membranes and expose their protein content.
So in order to isolate the protein content from the bacterial cell, we then perform a methanol chloroform water precipitation. And once we have these proteins isolated, we then go ahead and add a pronase. This pronase will break down our proteins into its individual amino acid components. And then we can run an ultrafiltration and or chromatography in order to purify the amino acids. We can then run our samples through the LTQ Velos Pro, which will allow us to interpret the data. So a little bit of how this data is actually created is our samples need to be ionized first. So our LTQ Velos can then separate the ions based by molecular weight. We then get a bunch of different readings and we interpret the average.
On this graph, you can see each peak represents a molecule with a specific mass. Each red letter designates a specific amino acid. We're able to identify each amino acid because each one has its own unique mass. 174.5 corresponds to arginine, 132 corresponds to leucine, etc. We were able to see each amino acid we expected to find on our E. coli sample.
So what does this all mean? Well, this means that the protocol we actually helped develop does in fact work, which is nice. And we were also able to prove that the pronase was able to digest other proteins such as bovine serum albumin and apple myoglobin.
During this project, we determined that it's possible to engineer proteins with special properties, and the results of our data can be applied to future research in multicellular organisms or mammalian tissue, including human cancer cells.
And this concludes our presentation. I hope you guys enjoyed.